Hello everyone, my name is Jessica Charles and I will be discussing microbial diversity and antibiotic production of treehole ecosystems. While most antibiotics come from soil environments, there are many other environments that also host diverse communities. The research in my advisor's lab focuses on mosquito breeding habitats, and we were interested in what type of microbial communities are growing in treeholes and whether there are any antibiotic producers present. The collection of water from tree holes was an interesting component of our research since most of my lab's work focuses on the mosquito communities and also because soil is typically collected for these types of microbial analysis. The objective of this project was to identify antibiotic producers from tree hole environments, which are common mosquito breeding habitats. Water was sampled from 2 tree holes, shown in Figure 1, in New York every 2 weeks for a total of 8 weeks in September and October. These tree holes are part of a long-term mosquito monitoring program. I diluted the samples from 1 to 10 to 1 to 10,000 using 2 types of complex media. Figure 2 is an example of one of the sets of serial dilutions that I performed. From these serial dilution plates, I calculated colony forming units for each collection. Afterwards, I plated 138 isolates against Escherichia coli, Staphylococcus aureus, and Pseudomonas aeruginosa, which are 3 pathogens widely associated with antibiotic resistance. From there, I conducted PCR and sequencing in order to identify and compare them to GenBank sequences. As for results, I did not find a difference between the number of colony forming units across the 2 tree holes, time of collection, or media type. As you can see in Figure 4, we notice an upward trend of colony forming units as time goes by in the season. However, this wasn't significant. I screened 138 isolates against the 3 escape pathogens and 10 displayed inhibition against S. aureus. However, there was also no significant difference in the percentage of antibiotic producers found across the 3 variables. 6 isolates that demonstrated antibiotic production were analyzed with 16S rRNA sequences, and these showed sequence similarity to 3 bacterial genera and 1 family, all of which have species found in soil or water habitats. Based on the literature searches I did not find any information on whether these are known antibiotic producers. In conclusion, I was able to analyze the microbial community within 2 different tree holes and isolate antibiotic producers. Even though I didn't find a difference in the number of CFUs or percent antibiotic producers across the different trees, they are still potentially a source for novel antibiotics because the taxa belonging to the antibiotic producers that I found are not well studied in terms of antibiotic production. This study provides an initial baseline for microbial communities found in treehole habitats. In order to expand on this research, future studies could collect more samples over a larger geographic range throughout the year, which would allow for a more thorough analysis of potential relationships between the number of colony forming units and antibiotic production in relation to these external variables. Thank you very much.